Picture a South American rainforest. Somewhere in the Amazon basin, roughly 50 million years ago, long before any human ancestor had developed anything resembling a complex brain, long before the first stone tool was ever struck, long before anyone had planted the first deliberate seed in deliberately prepared soil. A small ant is carrying a fragment of leaf. Not to eat it directly, not to build with it, she is carrying it underground into a network of chambers that her colony has excavated with the combined labor of thousands of workers to a specific room where a living organism is being cultivated, tended, fed, protected from competitors, and harvested for the nutritional output it produces in response to being cared for. The ant is a farmer. Her colony has been farming for longer than human beings have existed as a species, and the agricultural system she is participating in is, by almost every meaningful metric of agricultural sophistication, more advanced than anything human farmers achieved for thousands of years after the Neolithic Revolution. Let that land for a moment. The question of whether ants discovered agriculture before ancient humans is not a trick question designed to produce a surprising answer through semantic sleight of hand. The answer is simply yes, by approximately 49 million years. Fungus farming ants developed agriculture roughly 50 million years ago. Human beings developed agriculture roughly 10,000 years ago. The arithmetic of priority is not close, but the more interesting questions: what kind of agriculture ants developed, how sophisticated their agricultural systems actually are, what parallels and divergences exist between ant agriculture and human agriculture, and what the 50 million year head start has produced, those questions have answers that are considerably more extraordinary than the simple priority question suggests. Because the ant agricultural system, examined in full detail, is one of the most remarkable biological achievements in the history of life on Earth, and most people have no idea it exists at the level of complexity it actually operates at. Here is the complete story. Start with what fungus farming ants actually are, because there are more of them, more diverse in their agricultural practices, and more broadly distributed than most people realize. The fungus farming ants belong to the tribe Atine, within the subfamily Myrmicinae, and they represent a monophyletic group, meaning all fungus-farming ant species descend from a single ancestral population that made the transition to fungal cultivation roughly 50 million years ago in South America. From that single evolutionary origin, the atine ants have diversified into approximately 250 known species, distributed across the Americas from the southern United States through Argentina, occupying an extraordinary range of habitats from tropical rainforest to semi-arid scrubland to cloud forest to temperate woodland. All atine ants farm fungi, but not all atine ants farm the same fungi, and not all atine ants farm with the same level of agricultural sophistication. The tribe shows a gradient of agricultural complexity that ranges from the relatively simple practices of basal atine genera to the extraordinarily sophisticated monoculture agriculture of the leafcutter ants. And examining that gradient reveals something important about how agricultural sophistication develops over evolutionary time. The basal atines, genera like Myrmecocrypta, Apterostigma, and Scyphomyrmex, practice what researchers describe as primitive fungal cultivation. They grow fungi from the group Leucoagaricus on substrates including insect frass, dead plant material, flower parts, and similar organic material collected from the environment. The fungal strains they cultivate can survive and reproduce outside the ant nest. They are not entirely dependent on ant cultivation for their existence. This agricultural system, while clearly intentional and maintained, is relatively generalist, with different colonies sometimes cultivating different fungal strains, and with the agricultural relationship being less tightly co-evolved than in more derived groups. Moving up the sophistication gradient, the higher atine genera, including Trachomyrmex, Sericomyrmex, and related groups, show progressively more specialized agricultural practices, with more specific associations between ant lineages and fungal cultivar lineages, more elaborate garden management behaviors, and more developed co-evolutionary relationships between the farming ants and the organisms they farm. At the apex of atine agricultural sophistication sit the leafcutter ants, the genera Atta and Acromyrmex, whose agricultural practices represent the most elaborate, most productive, and most tightly co-evolved agricultural system outside of human civilization. The leafcutter ants are the farmers most people have seen footage of. The spectacular columns of workers carrying leaf fragments many times their body weight back to the nest, the enormous underground colonies with chambers specifically designed for different agricultural functions, the fungal gardens that are the heart of the entire colonial enterprise. The fungal cultivar of leafcutter ants belongs to the genus Leucoagaricus, specifically to strains that have been under continuous cultivation by leafcutter ant lineages for millions of years, and that, in the most derived cultivar strains, can no longer reproduce sexually. They have lost the ability to produce the fruiting bodies, mushrooms, through which free-living fungi reproduce. The cultivar exists only in cultivation. It is as thoroughly domesticated as any crop plant that human agriculture has produced, and the domestication happened through a process of mutual coevolution across millions of years of agricultural practice. This domestication detail is worth pausing on because it mirrors one of the most fundamental features of human agriculture. When human farmers domesticated wheat, maize, rice, and other crop plants, those crops changed under the selective pressure of cultivation in ways that made them better agricultural organisms and simultaneously less viable as independent wild organisms. Domesticated maize cannot disperse its seeds without human assistance. The husks that human selection produced to increase yield also prevent natural seed dispersal. The organism became dependent on the farmer as the farmer became dependent on the organism. The leafcutter ant's fungal cultivar has undergone an analogous transformation. The inability to reproduce sexually, the loss of fruiting body production, means the cultivar cannot maintain itself in the wild. It requires ant cultivation to reproduce vegetatively, spread to new locations, and persist across time. The ant and the fungus are locked in a dependency relationship that millions of years of coevolution has made essentially inescapable for both parties. Now examine what leafcutter ant agriculture actually involves at operational detail, because the summary description of ants that farm fungi dramatically undersells the complexity of what is happening in a mature leafcutter colony. The above-ground component of leafcutter foraging is the most visually dramatic element and the one most people have seen. Forager workers, one of the larger worker size classes in the highly polymorphic leafcutter caste system, locate suitable vegetation, cut fragments of leaves, flowers, or other plant material using their mandibles, and carry those fragments back to the nest along established trail systems that can extend 100 meters or more from the nest entrance. The trails are chemically marked with pheromones and physically cleared of debris by trail maintenance workers, representing an infrastructure investment that keeps the agricultural supply chain running efficiently. But the leaf cutting is not the farming. The leaf fragments are not food. The ants cannot digest cellulose and do not eat the leaves directly. The leaves are agricultural raw material, feedstock for the fungal garden. The farming happens underground. Inside the nest, the agricultural processing begins immediately when leaf fragments arrive. A second worker size class takes the incoming material and begins processing it, chewing it into a pulpy substrate, removing surface compounds that might be toxic to the fungal cultivar, and inoculating the processed material with fungal mycelium taken from existing garden sections. The processed leaf paste is then incorporated into the growing garden in a form that the fungus can colonize and use as a growth substrate. Smaller workers, the minims, the tiniest size class in the leafcutter worker spectrum, tend the growing garden continuously. They weed it, removing competing fungal species and bacterial colonies that would otherwise overgrow the cultivar. They groom the fungal hyphae, removing dead material. They apply antimicrobial secretions from their metapleural glands to suppress pathogen growth. They respond to areas of garden showing signs of stress or competitive invasion and concentrate weeding effort accordingly. The garden maintenance is constant, active, and sophisticated in its responsiveness to garden condition. The fungus, in exchange for this cultivation, produces specialized structures called gongylidea, small, swollen hyphal tips that are nutritionally enriched with lipids, glycogen, and free amino acids, and that appear to be produced specifically as a food reward for the ants. Gongylidea are not equivalent to the rest of the fungal mycelium. They are distinct structures with distinct nutritional profiles, and their production appears to be upregulated in response to ant-tending behavior. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. The fungus is, in effect, producing a crop specifically calibrated for its agricultural partners. And the agricultural partners are tending the crop in ways that stimulate greater crop production. This is reciprocal agricultural mutualism at a level of sophistication that took human agriculture thousands of years to approach through trial and error. The leafcutter ant and its fungal cultivar have been optimizing this relationship for millions of years through the mechanism of coevolution, and the result is a system of mutual dependence and mutual stimulation that operates with an efficiency no human agricultural system of comparable antiquity could match. The colony scale at which this agriculture operates is itself remarkable. Mature colonies, the largest leafcutter species, can contain 5 to 8 million workers, with the fungal gardens they maintain spread across hundreds of underground chambers occupying a nest volume of tens of cubic meters. The daily leaf harvest of a mature colony is measured in kilograms, substantial enough that leaf-cutter ants are significant herbivores in the ecosystems they inhabit, removing leaf material from the surrounding forest in quantities that influence local plant community composition and dynamics. A single large colony harvests more leaf biomass per day than many individual large mammalian herbivores. The agricultural productivity enabled by fungal cultivation, which concentrates the nutritional value of difficult-to-digest plant material into the easily digestible gongylidea produced by the cultivar, allows leafcutter colonies to support population densities that would be impossible if the workers were eating the leaves directly. The pest and disease management practices of leafcutter ant agriculture deserve specific attention because they represent one of the most sophisticated elements of the agricultural system and one that draws the most direct comparisons to the challenges of human monoculture agriculture. Monoculture agriculture, the cultivation of a single crop variety across a large production area, is vulnerable to pathogen outbreaks in ways that more diverse agricultural systems are not. When the same genetic substrate is present everywhere, a pathogen that can infect it has unlimited area to spread through without encountering resistant hosts. Human monoculture agriculture has learned this lesson repeatedly through catastrophic crop failures caused by fungal diseases, bacterial pathogens, and insect pests. And managing these risks through pesticide application, crop rotation, varietal diversity, and biological control is a major ongoing challenge. Leafcutter ant agriculture is monoculture agriculture. A single fungal cultivar strain is maintained throughout a colony's garden, clonally propagated by vegetative division rather than sexual reproduction. The vulnerability to pathogen invasion that this creates is real and has driven the evolution of sophisticated pathogen management systems that the ants deploy continuously. The primary pathogen threat to leafcutter fungal gardens is the parasitic fungus Escovopsis, a specialized pathogen that attacks the Leucoagaricus cultivar and can devastate gardens if not controlled. Escovopsis is adapted to the garden environment and can spread rapidly through the monoculture substrate if the ants' defenses are compromised. The ants counter Escovopsis through multiple mechanisms deployed simultaneously. Worker grooming removes Escovopsis spores from garden surfaces before they can germinate. Metaplural, gland secretions, an antibiotic compound produced by glands on the thorax, suppress Escovopsis growth. Garden waste material, which has elevated Escovopsis spore loads, is removed from the garden and deposited in dedicated waste chambers located at the periphery of the nest, preventing contamination recirculation. The most remarkable element of leafcutter ant pest management involves bacteria. The ants maintain populations of pseudonocardia bacteria, actinomycete bacteria that produce antibiotic compounds specifically active against Escovopsis, on specialized regions of their cuticle. The bacteria grow on the ant's body surface in a cultivated patch fed by exocrine gland secretions that provide nutrients for bacterial growth and producing antibiotics that the ant applies to the garden through grooming and contact. This is a three-level agricultural system: ants cultivating fungi, ants cultivating bacteria to protect the fungi, and bacteria producing antibiotics that suppress pathogens. The leafcutter ant is simultaneously a fungal farmer and a bacterial farmer, maintaining two separate cultivated organisms in a nested agricultural arrangement that serves the primary fungal cultivation enterprise. The Pseudonocardia Escovopsis leucoagaricus system has been co-evolving for millions of years in an ongoing evolutionary arms race. Escovopsis evolves resistance to the antibiotics produced by Pseudonocardia. Pseudonocardia evolves to produce novel antibiotic compounds that overcome the resistance, and the ants maintain this bacterial defense system that provides some protection against any given Escovopsis strain while the evolutionary arms race continues. Research into this system has produced genuine interest from pharmaceutical researchers because the antibiotic compounds produced by ant-cultivated Pseudonocardia strains represent a diverse library of compounds with potential medical applications. Natural products produced by organisms that have been under selection for antibiotic efficacy for millions of years. The queen's role in agricultural continuity deserves specific examination because it represents one of the most elegant solutions to a fundamental challenge in any agricultural system: how to transmit agricultural knowledge and biological material across generations. When a leafcutter queen leaves her natal colony on the nuptial flight, mates with multiple males, and prepares to found a new colony, she does not leave behind the agricultural system. She carries it with her. Inside a specialized pocket in her mouthparts, the infrabuccal pocket, she carries a small fragment of the fungal cultivar from her mother colony. This founding inoculum is the agricultural seed stock for her new colony. When she excavates her initial founding chamber and begins laying eggs, she also introduces the cultivar fragment and begins cultivating it with her own secretions until her first worker offspring can take over garden management. The transmission of the cultivar from mother colony to daughter colony through the founding queen is the mechanism by which agricultural continuity is maintained across the splitting and founding events that propagate leafcutter colonies through landscapes. Every colony traces its cultivar lineage back through a chain of founding queens to ancestral agricultural origins. The cultivar has been propagated through this mechanism, without sexual reproduction, for millions of years. A continuous agricultural tradition maintained across a time span that makes the entire history of human civilization look like an afternoon. The comparison to human agriculture illuminates both the parallels and the profound differences between the two agricultural systems. Both human agriculture and ant agriculture involve the deliberate cultivation of specific organisms in controlled conditions, the active management of pests and competitors, the development of infrastructure to support agricultural production, and the organization of labor around agricultural tasks. These parallels are real and striking, and they reflect the fact that agriculture as a strategy, concentrating food production through controlled cultivation rather than foraging, represents a discoverable solution to the challenge of supporting large, dense populations that convergent evolution arrived at independently in two very different lineages. The differences are equally instructive. Human agriculture is characterized by rapid innovation, the development of new techniques, new crop varieties, new tools, and new approaches to pest and soil management across historical time. Leafcutter ant agriculture is characterized by extraordinary stability. The basic agricultural system has been operating in recognizable form for 50 million years with relatively modest innovation across that span. The stability reflects the evolutionary rather than cultural transmission of agricultural knowledge in ant colonies. Innovations are incorporated slowly through natural selection rather than rapidly through social learning and deliberate experimentation. Human agriculture has transformed landscapes, climates, and ecosystems at a global scale within 10,000 years. A pace of ecological impact that reflects the cumulative culture advantage discussed in other contexts. Leafcutter ant agriculture has been operating for 50 million years and has certainly shaped the ecosystems it participates in, but the pace of ecological transformation is fundamentally different from what human agricultural expansion has produced. The diversity of human agricultural systems, the thousands of crop species, the range of cultivation techniques, the adaptations to environments from Arctic to desert to tropical, reflects the flexibility of cultural innovation applied to local conditions. The diversity of atine agricultural systems, the gradient from primitive to sophisticated cultivation across 250 species, reflects biological diversification across evolutionary time rather than cultural innovation within a single lineage. What the comparison most clearly establishes is that agriculture, properly defined as the deliberate cultivation of another organism for food production, is not a uniquely human achievement. It is a strategy that evolved independently multiple times in the history of life, not only in ants, but in certain beetle lineages that cultivate fungi, in certain termite lineages with fungal gardens, in damselfish that maintain and defend patches of algae, and in various other examples of what biologists broadly call farming behavior. The atiny ants are by far the most elaborate and most sophisticated non-human agricultural system, and their 50 million year priority over human agriculture is real and remarkable. But what human agriculture added to the concept of farming was something the ants, for all their agricultural sophistication, have not achieved: the capacity for rapid, culturally transmitted innovation that has allowed human agricultural systems to transform in decades what ant-agricultural systems transform across millions of years. The ant sitting in the opening scene of this account, carrying her leaf fragment into the underground chamber where the fungal garden waits, is participating in an agricultural tradition 50 million years old. She has no awareness of this tradition. She has no concept of what farming is or that she is doing it. She is following behavioral programs encoded in her genome and expressed through the chemical communication and developmental differentiation of her colony, programs that were shaped by 50 million years of selection for effective fungal cultivation. And the system she is participating in works. It has worked without fundamental redesign for longer than most species that have ever existed on Earth have existed at all. The fungal garden her colony maintains will produce the gongylidea that feed millions of nestmates. The colony will grow, fragment, found new colonies, and propagate the agricultural tradition across another generation. 50 million years before any human planted the first deliberate seed, the ants were already farmers. They were very good at it then. They are very good at it now. They will probably still be farming long after whatever comes after us has stopped being interested in the question.